I can tell you one of the remarkable things for me was within 1 week when I diagnosed a 72-year-old with Rett syndrome and a 3-week-old with Rett syndrome, neither one of which most of my colleagues had ever done. Can you believe that? These people are, sort of sitting in plain sight, so to speak.
When Evie started missing her milestones, we began the diagnostic journey to find out what was happening. For 5 heartbreaking years, we chased doctors, developmental pediatricians, neurologists, and metabolic specialists, scheduling blood work, MRIs, EEGs, microarrays, and still, no one could tell us what was wrong with our little girl. It wasn't until we did a whole exome sequencing that we finally got an answer. A point mutation on a gene called SHANK3, also known as Phelan-McDermid syndrome. Our story is painfully common for families living with genetic disorders. The emotional toll is immeasurable, and the cost to families and the medical system is in the tens of thousands. But we may be the last generation of parents to suffer that indignity. Whole genome sequencing is changing how we diagnose newborns. Instead of years of guessing and testing, we can get a full picture of a baby's genetic health early in life and move straight towards potential treatments. Or at least to a community and a name for what's going on when no treatments yet exist. Dr. Wendy Chung leads GUARDIAN, a newborn genome screening study that's testing what it would mean to bring this technology to families. In this episode, we talk about what her team is finding and the challenges of moving from today's newborn screening programs to a future built on whole genome sequencing. Hi, I'm Ron Clayman, and this is Generation Hope. I'm at the New York Academy of Medicine for the ICONS Conference, the International Consortium on Newborn Sequencing, where experts are exploring what happens when genome sequencing becomes part of routine newborn care. While I'm here, I sat down with Dr. Wendy Chung, a clinical and molecular geneticist, chair of pediatrics at Boston Children's Hospital and Harvard Medical School, and principal investigator of the GUARDIAN Newborn Genome Screening Study. If conversations like this are helpful, please subscribe, like, and share this video so more families, clinicians, and researchers can find it. And now, Dr. Wendy Chung.
I'm Wendy Chung. I'm the chair of pediatrics at Boston Children's Hospital and Harvard Medical School, and I'm a pediatric medical geneticist, and so I think a lot about rare genetic conditions.
Okay, wonderful. I want to start really early on and really focus on this question is really about you. I want to know how you got into this. What inspired you to do what you do?
So I knew early on that I wanted to be a scientist. I started out thinking about biochemistry from a very basic point of view, basic biology point of view. And I spent, during one of my summers as an undergraduate, spent time at the National Institutes of Health with, at the time, Seymour Kaufman, who was studying phenylketonuria. And when I did that, that's an inborn error of metabolism with a biochemical defect that makes people potentially, if they're not caught, diagnosed, and treated, can lead to intellectual disabilities. But with early diagnosis, even as newborns, we can put them on a special diet, which is depleted in phenylalanine, and they can actually grow up healthy. And with that, I entered it from the point of view of being a biochemist, thinking about the biochemistry angle. But that summer, I realized that I had a lot to learn, which is not surprising. And I was fortunate to be able to meet patients in the hospital at NIH and be able to understand what their life was like. And also began at the time to start exploring use of genetic tools to be able to make the diagnoses for people with atypical PKU. And that taught me several things. Number one is I realized I really liked patients and I really wanted to be then a physician scientist to understand the aspects of the underlying science, but also how it applied to people. I also fell in love with genetics. And then the third part about it was it started this lifelong journey to try and think about early diagnosis for treatment to be able to have the maximal impact. And that really has been, unfortunately, now that I think about it, about 35 years ago in terms of starting that journey. And it's taken some time for the tools to be developed in terms of human genetics with the Genome Project, some of the tools now in therapeutics. But my hope is that I'm still giving myself another 20 years until I retire. But I would like to be able to retire by having a very different world for children in the future. I hope that by that point we will be able to, in a very big way, think collectively about rare genetic conditions and how we can get everyone, and I really do mean everyone, every child, every newborn, access to early diagnosis and early treatment. And so that's, I hope, a different world that I hope the next generation will grow up living.
Amazing. And without being too overly optimistic, realistic, do you think it'll take 20 years?
There are some famous people, Bill Gates among them, that will say that they can't necessarily precisely predict the timing of certain things, but that generally they're enabling technologies which will propel us forward. And I do think that's absolutely true when it comes to genetics. I think in terms of the diagnostic side, the screening side, it's not a matter of if but when, and potentially there are ways of moving that forward relatively quickly. I think in the diagnostics, they're going to be, I call it mathematically a step function. So in other words, it's not as if we're going to linearly increase one by one, but they're enabling technologies that will allow us to bring forward platforms, two parts of the body to do this, which all of a sudden it won't be just one condition, but hundreds of conditions that will potentially move forward. And what I can't time is the timing of that step. But once that step occurs, I do think it'll be important. And I think a lot about collectively, how can we all prepare so that when it's time for that step to occur, we're ready to go and we don't lose a second as soon as we're ready.
So these types of things you're talking about, is it like AI, quantum computing, founding of CRISPR a decade ago or so, like all these types of things that come together at a particular point that accelerates?
So there are a few things that come together. I'll just take one technology. So let's pretend that one knew what the target was in terms of what the gene was and specifically what the mutation was. Let's pretend we now have ways of going in and erasing the genetic variant that causes the disease and writing in the correct genetic variant, a method of gene editing. So that's one of the technologies. Can we do that in terms of being able to do that and do that across, let's say there are 10,000 rare conditions, rare genetic conditions, and that within that, any one gene might have probably not just one genetic variant, but a multiplicity, maybe even 100 or hundreds of genetic variants. Once one has the technology to be able to target the right gene and target the right place, that's one enabling technology. And I want to be very clear, we don't want to disrupt any other genes in the process. The next is to be able to get to the right part of the body, the right perhaps part of the body or even cell type within the body, and having an address, a homing way of being able to get to that cell. And so that's location. And then the third component is time. So to be able to get into the body, into the right cell at the right time. In general, I think it's going to be the earlier the better in general. But on the other hand, there is this concept of just in time. That is that if you don't need to treat too early and there's a chance of some toxicity or something going on, you don't want to do it superfluously, but you want to be able to do it just in time. Technologies or platforms like newborn screening would allow us as a public, from a public health and a societal point of view, to get to everyone. But that may be, sadly, even too late for some conditions. And so I hope it's not most conditions, but I'll even throw out the concept that perhaps even while mothers are still pregnant, for some conditions, we might need to even, from a timing point of view, get in then. Just to say that I don't want to leave anyone behind as we think about this. And so we are even thinking about how do you make the diagnosis even earlier for some conditions, and how do you get to the right part of even a fetus in terms of fetal therapy. With all of those three components, I think those three enabling technologies or ways of doing things. But I can see how we're going to get there. There are emerging technologies to do that. And there are a lot of people working on this very hard.
Quick little break here. Sorry to jump in. But if you're finding this conversation helpful, please subscribe, like, and share. It might seem small, but it genuinely helps us make more content like this and tells YouTube to show these stories to more families, clinicians, and researchers. All right, back to the interview.
So for the gene therapy aspect of that, you're talking about base editing in terms of changing within the body, or?
So from a very technical point of view, there are three different technologies one can think about, or even more than three, but one can think about a gene addition strategy. One can think technically about a base editing strategy, that erasing and writing in. But there's some more generalizable forms of editing called prime editing. Ways of being able to get not even just one base, but potentially regions of a gene and being able to replace those so that it's closer to one size fits all, or at least a few sizes for one gene to be able to take care of many people. Nuances, but that's the basic idea, is being able to do that. And there may be some shorter term, what I call bridges, until a more permanent solution. So people are thinking about things like ASO strategy, like RNA editing or RNA delivery, and some of the same technologies, for instance, that many of us get vaccines now these days with. So a variety of different modules that one could use for these. But hopefully between some of these as a bridge that may require repeated dosing, eventually we get to what I think of as a one and done solution.
What do you see as the biggest challenge with early diagnosis right now? Is it political or is it the actual science? It seems we could do it. It's just whether the cost comes down enough to a point where I'm getting ads on Facebook now from sequencing.com because of my search history.
Yeah.
So what is the biggest hurdle that you see?
I've been thinking about this, as you can imagine, really for a long time. So we had started the first pilot study for newborn screening with this paradigm of early diagnosis was necessary for early treatments in this window of opportunity. So this actually started in 2016 for me, at least with spinal muscular atrophy. So we did this, and it's a DNA-based diagnostic. In this particular case, very focused, just looking for the gene survival motor neuron that's related to SMA, spinal muscular atrophy. And with that, for those of you who don't think about this all the time, babies with SMA when they're born initially look perfectly fine, but it's a degenerative process. I'll come back to that, but a degenerative process such that by the time you clinically see that a child is having problems and diagnose them, those what are called motor neurons have died, and that is probably an irreversible process. And the idea that I had for this was you needed to be able to, even in clinical trials, that the best responding babies or children in clinical trials would be those treated pre-symptomatically, because otherwise, once you went downhill, the most you might be able to do is stabilize. Whereas if you could stabilize at a normal function, that clearly would demonstrate efficacy. So the idea became leave no one behind is a theme that I really care a lot about. And so we use the newborn screen blood spot that's collected anyway. I'll go back to PKU because we started out doing it for phenylketonuria. And so it was no additional sort of difficulty for babies, no additional pokes, sticks, anything else. And we had have an infrastructure for newborn screening. And so every single baby is part of this process. It's a way to ensure that no baby's left behind. So we started that screening study in 2016. 93% of parents who were offered that said, yes, I'd like to do that. No cost to them, but it was a consented research study. And in fact, the assay worked very well. We got the cost down. You mentioned the cost. We got the cost down of that one targeted assay down to less than $1 per baby. And as we did that, we identified children who otherwise would have been expected to have what is the worst form of SMA, type 1 SMA, where they usually don't live to their first birthday. And within that, instead, I have a little girl who's now in elementary school, but she went into the clinical trial at the time, and instead of dying by her first birthday, I have her home videos, she's walking almost by her first birthday, strong, never needed a ventilator to help breathe, never had any of the other problems. And so that, to me, was the birth for me of this, we can do this in terms of newborn screening as a platform. That was very interesting because reciprocally, we went back and forth very quickly. I want to emphasize that, from a pilot study of newborn screening to a clinical trial of a treatment in 2016 to within 3 years having actually FDA approval for 3 therapies and having that added nationally to newborn screening. So transforming completely in a very brief period of time what used to be, past tense, the most common genetic cause of death for children under the age of 2. So that was a demonstration project to me of if we can do it for SMA, what else can we do? So subsequently, we've done Duchenne muscular dystrophy. There's now actually an FDA-approved genetic therapy for Duchenne muscular dystrophy. We still need to get it officially added to newborn screening, but we had a very successful pilot study of that. And at that point, having done 2 of these and seeing how much effort it took, I got impatient. I tend to be that type. I said, rather than one by one, can't we do this dozens by dozens or hundreds by hundreds? And to your point, the cost of sequencing was coming down significantly. So the vision became, can we still take that same dried blood spot from newborn screening, and instead of doing a targeted test that was very inexpensive but very focused, can we offer the flexibility by having whole genome sequencing, sequence all 20,000 genes? We don't have to look at them all. We don't have to report them all out, because we don't even know what all of them do. But you have the infinite flexibility and adaptability that certainly as soon as a new medication is FDA approved, or even perhaps, and I say perhaps because we haven't done this yet, if a clinical trial were open, then you could start to identify those babies like we did for SMA. And I'll come back to that because there's one that I'm holding my breath for right now. So I didn't know whether that would work. So we're always very rigorous and evidence-based as we did this. The first thing that I didn't know is, could we technically do it? Could we use that dried blood spot? Would we be able to read out all of the data? Which genes should we report on? Would it work in terms of interpreting the data? Would parents even want this? So the first thing I did was I started with the parents who were part of that SMA study, and I asked them, literally, what do you want? Tell me in terms of, if you could design this, what tests would you want us to do on your baby? Then they gave me, after a lot of discussion, they gave me these constraints of, we want something that's a serious condition affecting children, young children. We operationalize that to children less than the age of 5. We want you to be almost certain. You don't have to be perfect. And almost certain for them ended up operationalizing to 90% probability that their baby would have this. Don't give them garbage, give them real information. And for sure, give them what I call news you can use, information where there's a treatment off the shelf immediately available. Those, slam dunk, no brainer for those. But then we had interesting discussions a la some of the conditions you've been talking about. So Rett syndrome in particular that I'll come back to. And families asking and thinking about other conditions associated with epilepsy in particular, again, back to the conditions you were talking about. And they said, you know what, and there was not uniform agreement, but they said, you know what, seizures are pretty serious. And if we could know that our baby was having seizures and treat those seizures before they were doing damage to the brain, that would, even if that's not a cure with a capital C, being able to do that and helping our child on the path to a healthier life, that's important for us to know. Until the time where there's perhaps a real cure with a capital C, but even just being able to recognize and treat seizures are important. So based on that, we included what I call an optional module. Parents don't have to do it, but if they're part of GUARDIAN, this genomic uniform screening against rare diseases in all newborns, this research study, they can opt into having this particular group of conditions, which started out as about 100 conditions. And as we did that, would have the ability to add conditions as there were new treatments approved. Fast forward, we've now done about 13,000 babies in New York City. So number one, we've proven we can do it. Almost always, almost 100% success now in terms of doing it. We can do it quickly, not quite as quickly as I'd like to. So we can do it. We started out, it took us at the beginning about 6 weeks. We've now shaved that down to doing it in about 3 weeks. Ultimately, I'd like to be able to do it in about a week, but we're getting better as we're doing it. All free to parents, but it's still expensive on my end from a research point of view. So we need to continue to bring that cost down. And we're not perfect, but as we talked to families about this, we recognized that we weren't perfect, but that we would work with families through from screen, because this is just a screening test to start with. Like a mammogram is just a screening test. It doesn't diagnose a definitive diagnosis, but we start out with the screen. We walk people through to a diagnosis and we onboard them to care. So we go that entire pipeline. It's not just diagnose and adios, it's them the full way in terms of that. In doing that, we also have now realized that, number one, some conditions are more common than we realized. That's probably because there are patients who are never diagnosed. I don't know if they're not diagnosed because they die. I don't know if they're not diagnosed because they're symptomatic, but they just don't get to a genetic diagnosis in terms of a diagnosis. But we know that for some conditions, Rett syndrome in particular, the frequency is much higher when we do newborn screening, population-based screening, than it is based on who's symptomatic and goes and eventually gets a genetic diagnosis. So Rett syndrome, probably you already know this, but Rett syndrome is like SMA in the sense that the babies start out normal. They start out normal for the first about year, sometimes a little more than a year, and those girls then start to decline. And the big question that I have in my mind is, can we bend that curve so that those girls don't decline, but can we stabilize them at normal function if we could get the treatment into their brains early enough? So we put this on the list because, in fact, there is an FDA-approved treatment for Rett syndrome. It's not perfect, though. And even though there's an oral medication that you can take, it doesn't completely reverse the condition. It's does a little bit.
You're talking about
Tide.
Okay.
So it's a medication that's available. It's not perfect in doing this, but there is, and again, remember the timing, there is, as you said, an ongoing set of clinical trials available, not available to newborns, importantly. So available to individuals a little bit older to demonstrate safety and hopefully efficacy. In the meantime, and I have to tell you, I literally am holding my breath. I've got these little baby girls now that we have identified through Guardian, through the newborn screening. We're watching very carefully to see what their brains are doing, see how their development and their behavior is coming, and literally on the standby list, watching to see in terms of these clinical trials and when it's going to be safe to be able to bring it down to their age so that if their parents want to, a big if, but if their parents want to, then they'll have the opportunity, like we did for SMA, to see if these are going to help their daughters.
Is it also 2 to 9 years old for Rett in the trials?
So that's what the trial is. So it's not, none of our girls yet are 2 years old.
The work you did in terms of getting SMA on the panel, SMA was the 50th, right? The most recent thing added to the newborn screening panel?
I think there's been one more added since then.
Okay.
But,
so, and you were saying that you give parents the option of what they want to know. Was there ever the consideration of knowing of something that there is no treatment for and what the benefits of that are? I can speak from my own experience. There's no treatment for PMS yet, but for 5 years, we didn't know what was going on. We went from specialist to neurologist to metabolic specialist, developmental pediatricians, not knowing what was going on. So there's the aspect of, number one, you can stop looking for parents who do that. But then there's also it's like a catch-22. If you don't know, or you don't know there's a community out there to help answer a lot of these questions, but also to help raise awareness, raise research funds like CureShank and PMSF or RSRT, the benefits of knowing to come together and push the research and to where it needs to go.
Sure. I thought about this, and again, this was obvious from families like you very early on. So by virtue of the fact that we have whole genome sequencing and it's done in a clinical laboratory, what we call CLIA clinical laboratory, those data are retained in that clinical laboratory. And any child who becomes symptomatic, so outside of the newborn screening, but becomes symptomatic, that family has the opportunity to have those data reanalyzed now within the context of that baby or child's symptoms, to then ask the question, could there be a genetic condition that was not screened for, but that is someplace else in the genome? So I did that specifically because, again, I wanted to make sure every child has an access to a diagnosis, and I didn't want to limit it just to this that we were doing in Guardian, but allow all of the children with any condition to be able to access that. That, for me, initially has been the right balance in terms of being able to allow that flexibility and being able to give people access. But still, and I would love to hear feedback from the community in terms of this next issue. The thing that has been interesting to watch families now for a little over 2 years and guide them through this process, because like I said, I literally am with them from calling them to telling them what we found in Guardian through the process of continuing to onboard them to care, in some cases after that. And if you think about yourself as the parent of, say, a 3 or 4 week old baby that looks perfectly fine and that you have enormous hopes and dreams for, and then to be able to be given a very serious diagnosis completely out of the blue when your baby looks perfectly fine, that's hard. And part of it is if there's nothing more that you can do right then. And I'm not saying, and I totally know exactly what you mean about there's lots that you can do. You can support your child with early intervention. You can educate yourself. You can help them with feeding therapy. Like, I'm not dismissing that at all. But when there's not something that's transformational and immediately time sensitive, to balance that in terms of what some parents have thought of as, some parents have called me as the honeymoon for them, being to enjoy their child and have that joy that comes with that.
Yes.
As opposed to the heavy weight of that information when their child is not yet symptomatic. That's the balance that I've been trying to think through. And I don't know, I'll be very honest, I don't know the answer. And I've been thinking to myself, is it best to allow parents to choose their own adventure? And would I give parents the option of, if to know that information at all, And if so, when? And how many options would you give parents about when to know that? The easiest thing to do it is one single workflow. Everyone gets the information at the time we have it. We don't hold anything back. One easy stream. Is that ideal? Is that best? I don't know the answer. Would families like to have the information but know it at 3 months of age, 6 months of age, 12 months of age, only when their child becomes symptomatic? I don't know. And I would love to hear feedback.
I think you'll get a full gambit of answers. And I think that kind of points to it being, like you said, something that may be parent-driven.
Yeah.
Parents who want to know can know. Parents who want to be in the honeymoon phase, who let them be in the honeymoon phase until there's something, a sign that their kid isn't meeting the milestones and they want to find out why. I think it's not for anybody to make that decision for the parents other than the parents. Yeah.
So the good thing about exercises like this is I would love to have prospective parents have this seed planted so that they could think to themselves, rather than we come to parents, for instance, right after the baby's born. And if you think back, that can be the delivery, the new mother may be exhausted. They may be filled with all sorts of emotions
or drugs.
or drugs. And so that may be a hard time to process that information, right? So part of now this exercise is to allow prospective parents to think about it at a time when it's not so time pressured. And if they have the moment to think about it, think to themselves, would I like to know this? And if so, when? And then, like I said, know yourselves better than I do in terms of this. Can I support a workflow that's concordant with what your preferences are in terms of knowing this information? And that's partially on me, but I'd love to be able to hear what the answer is.
Yeah, you'll get a million different answers when you ask a million different people. You can certainly set up a system that allows a million different opportunities.
Maybe not a million, but a few.
The data from the GUARDIAN study, has it been published yet? Or are you exposing it to the world in the next couple of days?
So in the next couple of days, there's a meeting called ICONS, which is an international meeting of several of us doing newborn genomic screening studies. I'll be presenting at ICONS, and hopefully information will be published pretty soon.
You were talking about the prevalence of Phelan-McDermid syndrome. Same thing in all these genetic disorders is that nobody really has until now a real idea of what the real prevalence is. With Phelan-McDermid syndrome, they throw in round numbers, and they're 1 in 25,000 and so forth. Some of the researchers processed the data, and they were thinking that it's probably about 3 or 4 times the prevalence of what the numbers are showing now, simply because most people don't get a genetic diagnosis. With Rett, I'm guessing like the numbers are significantly higher than what's published.
Yeah. So Rett syndrome, certainly the number that we're seeing, which is still relatively small numbers, but about 1 in 1,500 girls, which is remarkably higher than what has been published.
So about 1 in 10,000?
1 in 10,000 girls is what's been published.
That's huge. So with that comes more eyeballs on Rett syndrome, more investment into Rett syndrome.
That's my hope. The realization that in terms of, pardon the expression, but disease burden, there are many more individuals affected by this than I think are diagnosed. And with this, many more girls that we could, and boys as well, but especially because this is an excellent condition, girls, individuals that we could be helping.
As part of the Facebook groups, we see people, hi, my daughter has just been diagnosed with Rett, or with the Phelan-McDermid syndrome. She's 47.
I can tell you one of the remarkable things for me was within 1 week, when I diagnosed a 72-year-old with Rett syndrome and a 3-week-old with Rett syndrome, neither one of which most of my colleagues had ever done. Can you believe that?
72 years old?
Yes. So one of the things that I, had a wonderful partner with a reporter, a journalist in Vanity Fair, and we went through her family story of an older relative who had intellectual disabilities and had been institutionalized, and always this question of what happened to her aunt. And so we went through that journey and did her genetic testing. And with that, a number of people have seen that particular article and come to me with the same question in terms of this relative, this sister, this someone else within their family. And as we've gone through and done genetic testing, it's remarkable that these people are sitting in plain sight, so to speak. For obvious reasons, they're not necessarily going to their doctors these days and asking for genetic testing. On the other hand, they're remarkably insightful to me for the generation of children and young people growing up now, because we have some sense of what their obstacles were during life, we can look backward now and be able to help the next generation growing up. So they're sitting there, and it's absolutely possible to diagnose them if they want to be.
To have this sense that this generation will be the last generation.
We can only hope.
Yeah. Yeah. The working title of this is Hope and Genes. So the GUARDIAN study, 13,000. I think on your website I read last was 10,000.
Yep.
So it's gone a couple of months more. And I thought it was in Boston, but you're just in Boston. It was in New York.
So I was at Columbia University for many years, and I started this study in New York City at the New York Presbyterian Hospital Networks affiliated with Columbia. So it started when I was still here. I've since moved to Boston, but I still have my team locally here in New York City, and we're still going strong with running the study in New York, and in all likelihood will spread to other locations as well.
It was all as newborns or children?
Newborns.
Newborns, okay. They wanted to be part of it, they checked off, and you had the full genome.
Yep,
sequence.
it's exactly. We use the same newborn screen, dried blood spots, so no additional heel sticks or pricks for the children. We go to their mothers after delivery before they go home from the hospital and tell them about the study. Then they have up to 30 days of birth to make a decision, but most of them do it prior to discharge from the hospital. And then if they agree to do it, which about 75% of them do, then we start doing the sequencing from that same residual dried blood spot and get their results back hopefully within a month.
Oh, amazing. Phelan-McDermid syndrome was not on that panel.
I actually have to look back because we've made a revision and update, what I call version 2.0, and I'd forgotten which conditions we were talking about, so I have to look and see.
I was going to ask you, what are the findings of the GUARDIAN study? Are you able to talk about what the findings were? Yeah, that'd be amazing.
As I said, the main things are that, number one, the majority of parents are interested in doing this. Even for this optional module that I described, of the 75% of parents that agree to do it, about 92% decide to do this optional module. So most people want as much information as they can in their minds to be able to help them take care of their children. We're able to do it quite successfully. We're able to do it either with a smaller number of genes when we did version 1 of this, or as we got confident we could handle it, we added more genes. So now we run about 450 genes. We're able to handle that just fine. And through that, about 3% of families get a positive screen result. So about 97% hear that everything that we checked is fine, but about 3% get a positive result. And then as we guide them through that next step, about 80% of the screen positives end up with a diagnosis where the screen ends up being a what we call false positive. And then as we take them forward, we're able to plug them into the right provider, sometimes a very specialized provider, sometimes it's just information their general pediatrician needs to know to take care of them. And many of them have started treatment based on what that diagnosis is now. And to my knowledge, all of them are doing well.
So the GUARDIAN study, you've got gNBS and Sanofi. They're sponsors of, they cover the cost of they do the sequencing, or?
So to be perfectly transparent in terms of who our partners are, so our partners are the New York State Department of Health in terms of who runs the newborn screening program. So they work with us very closely, along with our partners at New York Presbyterian Hospital. And then there are corporate sponsorship essentially from 3 sets of folks: gNBS, Illumina, and Sanofi. Illumina makes the sequencing instruments, so they help in terms of being able to provide some of the reagents that we use for sequencing. gNBS is the diagnostic laboratory that runs the sequencing, so they help us with that. And then Sanofi has a research grant to help us run the whole thing. They don't cover all the costs, but Sanofi contributes to covering part of the cost.
The hope is that this goes across the country, available through the gNBS, part of that system, and every hospital, baby's born, gets their little pinprick, and then that blood sample goes out through their state.
So I won't pretend to know exactly where this is going to go or the time frame for doing this. What I will say is that we're trying to collect the data that's necessary to assess whether or not the way we're doing it is right, whether as we do tweaks and refine the system, whether there are things that get her to a better way of doing this. And I won't presume I know the answer. What I can say from a feasibility point of view is it's possible to do, and families seem to be interested in doing it and happy with the results as we're doing it. what I think there is something called a recommended universal screening panel for the country, the entire country, about what gets included in newborn screening. There's a very regimented process that something goes through to be added to the RUSP. And if you were to talk about something being added across the country, it would need to go through the Secretary's Advisory Committee and be able to go through that process. Right now, there's not something that's gone through based on DNA sequencing alone. Spinal muscular atrophy is the closest that we've done so far, but nothing that's been on sequencing a complete gene, much less sequencing a genome or something like that. So we're in uncharted territory, I would say. That's why I'm not trying to be coy, but I just don't know because it's new. As we go forward, it certainly is going to require, and that's the platform, then each condition, at least the way it's set up now, each condition would get nominated and assessed based on the evidence review for that condition. And right now, you would have to identify at least one true positive baby through a pilot screening study to demonstrate the success of the screening method, be able to go forward. And so the reason why Guardian is doing what I think of as very large numbers is because across those conditions, we still need to identify at least one baby. And you can imagine some conditions are and truly are 1 in 100,000 or 1 in 500,000. So you've got to screen a lot of babies to get that one at this point. So within this, I don't know how long it's going to take us to get up to the number of babies we need to. I don't know how long it'll take us to go through the process of the RUSP and what their feedback will be. But as we're doing it, this does have scalability because you're not doing one demonstration project with 10,000 babies for one condition and then starting all over, right? There's an economy of scale built into this.
And there are other kind of research studies similar going on in different states.
So there are other people within this space of doing different types of things around the world. So not just in the United States, and different countries have different rules. So there are people in European countries, people in Australia, people in the Middle East thinking about this. And the interesting thing is none of us are exactly the same, either in terms of the genes that are studied, even the sample type that's being analyzed, the results that are being returned. Everyone is a little bit different. So we're learning from each other as we're doing it. In the United States, I fully expect that it will be not just data from one state, but probably data from either more than one study or at least more than one state in terms of demonstrating generalizability.
Are there any privacy concerns with insurance companies? For example, I remember when CRISPR came out and people were worried about the ethics of it, of insurance companies requesting information and refusing insurance based on genetic probabilities. Has that been dealt with in the U.S.?
Two types of privacy concerns. One is the question of, is there a chance of discrimination for the baby based on a genetic predisposition that hasn't been realized? And within this, you'll recall that we're returning information for conditions that should be symptomatic before 5 years of age. So when it comes to that, we are specifically not returning something that might be a BRCA1 or BRCA2 cancer predisposition when you're 50. That to me is not something that's relevant to the care of a newborn baby. There's not anything I'm going to do differently for a baby girl at the age of 3 based on that. And that's the type of thing I worry about, long-term genetic discrimination that isn't obvious in a little one. And so right now, there's nothing that we're returning that puts someone in jeopardy of that type of discrimination. There is an issue that people might not have thought of, but I'm going to surface it just to make sure we have the conversation publicly. So people do get concerned about privacy when it comes, even for instance, to law enforcement. So people might not have thought about this, but DNA is uniquely identifying for an individual. It also tells you about your family lineage. And so it also could tell you not just about you, but it could tell you about close relationships like your father or mother and people who have done Ancestry.com. In fact, that's how they find some of their relatives. So the concern has been raised about whether those dried blood spots or those data could be used by law enforcement potentially to implicate a family member, for instance, in a crime at which there's DNA evidence. And I do think longer term, to give society reassurance that this will not come back to bite them in some unintended way, we'll probably have to legislatively deal with the privacy concerns in that way.
Seems like everything that happens now with technology, the legislation follows way behind.
It's with new technologies and new data sources, it takes time to catch up, and legislatures are much slower. They're not nearly as nimble as science is.
I can't express how excited I am to hear about things like the Guardian study and Project Findouts, it seems to me that everything is accelerating at an exponential rate. What you were saying about the different things that are coming together and how that's going to accelerate things, and it's like a step up. It certainly feels things are really coming together. If you step back and seeing all the things that are happening together, it's very exciting.
Well, it's definitely, I think I try not to overhype things, but I do want to say that I really do have hope. I truly do, like in my belly, feel this hope. And as I do this, I'm trying to collectively think for all of us about 3 steps ahead of what we're going to need to be able to make sure that goes as quickly as possible, not reckless, but as quickly as possible and as inclusively as possible so that we're not leaving anyone behind as we go forward.
In my conversation with Alex earlier today, I asked him, have you sat back and played it through and thought about what it means if these gene therapies work? Actually, you have kids or people varying ages and their brains come online. So what does that look like?
Yeah.
I've gone through that process.
Yeah.
And it just blows my mind each time in that I don't think anybody's preparing for that yet. I'm curious if anybody is thinking 3-4 steps ahead on these intellectual disabilities and the gene therapies that are available for them. And what does that mean? And how do we get ready for that? How do we get families ready for that? How do we get the patients ready for that?
So I think as communities, I think we have a lot to do in terms of being lined up. That means, and I realize how busy and complicated families' lives are, but I say this in all sincerity, you have to stand up and be counted. And it's not very hard. Most of the research registries that are available, even if it's minimal viable data entry, you've got to have yourself registered and entered in those so that we count yourselves, so that as people go to try and get companies and research scientists to work on your condition, the more common it is as they go through these and prioritize them, the ones that are more common are the ones that rise to the top. So stand up and be counted. Some of these are mutation specific to the actual DNA variant, and you have to have your DNA variant registered. Otherwise, there's a possibility you'll get left behind. We have to know things like how old your child is, where they live. It affects things in terms of geographically, where we disperse things. And then in terms of figuring out what we're going to measure, what we're going to look at to see a change, I can tell you that knowing, because it's been asked for me in some of my N of 1 trials, how did you know that this child wouldn't have changed in the way you're seeing just without doing anything with a placebo? We have to know what that trajectory looks like without doing anything, and we can do that now. We can have that done and in the bag and know what exactly we would use in terms of outcome measures. And I don't want to be burdensome to families, but there are increasingly, you asked about data, AI, and being able to use everything. There are ways of getting denser data so that you can be more sensitive to change and be able to get an early readout if something's working out, requiring fewer individuals and knowing faster. And if we have to be flexible and nimble and iterate on things, we want to see early signs as much as we can. And so we may be using things that may sound funny, but I think it's best to get people in their home environment rather than bringing them into a hospital or clinic where kids may not be on their best day. They may not have gotten a good night's sleep. So I want to think about ways that we can collect the data for kids in their natural setting, where they usually are, behaving the way they usually are, get dense data so that we can see the change and be more inclusive so that people that maybe hundreds of miles from a site where we're doing a trial can also participate by doing it from home in a remote way. If we can think about doing that, I'm convinced that's the infrastructure we collectively in the rare disease community can and should start preparing now, so that as we get to the point where it's time to take that step, we are ready to go in terms of doing that. I'm going to use the term de-risks, but it de-risks it so that if we have to bring in a partner that may have more resources financially than many of us do in terms of rare disease communities, we hand it to them on a silver platter. We make it easy for them to be able to study our conditions. And within this, I don't a priori know which one of these conditions is going to be what I call reversible. I have hypotheses, I have best guesses, but you're right that we will be using data so that one rare disease will actually help other rare diseases, because we'll start to see patterns about which outcome measures work, which things are most sensitive to change, which conditions do seem to be most reversible, which ones do seem to be most amenable to certain treatments. If we collectively can think of ways of sharing that information, and to a certain extent, using AI to be able to see patterns that my feeble brain may be too tired to see late at night when I'm looking at the data, then we can be able to squeeze every little bit out of it to be able to either help our own children or help our neighbors' children as we go forward. So I'm optimistic that if we are intentional in this time when we have the time to think about it, we will be happy we've made that investment.
Like you mentioned, it's the burdensome aspect of it for families, especially families with special needs. It's just taking down data is such a challenge.
Yep.
I've tried it with my own daughter, just notes and stuff like that. And it just, you know what, you do it for a few days straight and life gets in the way. AI seems like a really good solution for that in terms of just getting unstructured data at the family level, like something, just talking to the AI, which you can just talk to it now, or put up a little video camera in your house and let it record everything and just send that to the AI. This is the patient, this is the mom, this is the dad, this is the, and so I can totally see how that would usher in a new level of data that we weren't able to get before, just simply because it was just too burdensome to get that data.
So exactly as you said, and obviously protecting individuals' privacies, letting families own that data and hold on to it until they're ready to do whatever it is they want to with that. But as you said, passive data collection, because life is just too challenging otherwise.
Yeah. I'm really excited to hear your talk. I'm going to show up there tomorrow at 7:30 in the morning and see. Just
walk in the door.
I'll do what I did when I was 13 years old, and I'll sneak into the movie theater. And then at the lunch break, I'll go and introduce myself, hopefully, and find them. Hopefully, I'll see you there as well.
Yeah, absolutely. Just introduce yourself as a family, a dad of a special person.
Yeah, I absolutely will. You've been fantastic. You've obviously done this many times. Yeah. But thank you so much. I really appreciate it. This is fantastic.
No, I'm really, like I said, I mean, I thank you because I'm intentionally trying to do what I said, which is that I really want to have families be thinking about what we need to do collectively now to be able to, whether it's for the next generation or for those who are born, and that we want to be able to improve their lives. But we can't miss any time and opportunity. So
thank you so much to Dr. Wendy Chung for sharing her work and great insight with us. Since we recorded this interview, there's been great progress. At the recent ICONS conference in London, the conversation around genomic newborn screening has shifted from "is it possible?" to steps to implement it responsibly in the real world. Pilot programs like PeriGenomed, BabySeek, Guardian, the Generation Study, Baby Screen Plus, and Baby Detect are showing that careful, hands-on experience is the key to scaling safely. The focus now is on scientific rigor, ethics, trust, and truly bringing families in rare disease communities into the decisions that affect them. We're also seeing real momentum from places like the UK, Italy, Abu Dhabi, Canada, the Democratic Republic of Congo, France, Saudi Arabia, Greece, and Ecuador, all taking different paths towards the same goal, giving babies and families answers much earlier in life. If conversations like this help you understand what's coming or give you hope for families like mine, please subscribe, like, and share this video so more families, clinicians, and researchers can find it. I'm Ron Clayman. Thank you for watching Generation Hope. And until next time, stay hopeful.